Welcome guys to my YouTube channel Real Science Simple, and let's make the science simple. Currently I am making lectures on the CRISPR. So today I'm going to cover the CRISPR-I and the CRISPR-A, which is the CRISPR interference and the CRISPR activator. So before moving forward, we need to understand what is the non-template and the template strand, because these are the things which you need to design your sgRNA. So this is the non-template strand, which is also known as the coding strand or the non-complementary or the non-target strand, and this is the template strand, which is also known as the non-coding strand or the complementary strand or the target strand. So why these things I am explaining here is because if you are designing your sgRNA against the promoter, then the transcription initiation is going to be inhibited, and if you are designing your sgRNA against the coding region, then in that case the elongation of the transcription is going to be inhibited. This is the same thing basically. So here you can understand, this is the sgRNA, and if it is binding to the promoter region, then the RNA polymerase cannot bind to the promoter region, and therefore the transcription initiation is going to be blocked. However, if you are designing your sgRNA against the coding region, then in that case this RNA polymerase cannot move, and therefore the elongation of the transcription is going to be blocked. So whenever you are designing your CRISPR-I experiment, your sgRNA should contain the seed region that will bind to the template strand. This is the dCas9 handle that will bind to the Cas9, and again this is dCas9, which is a dead Cas9. So both of the nuclease domains, which is the RUPC domain and the HNS domains, are mutated, so it can recognize the PAM sequence, but it cannot cleave the target strand. And this is your terminator sequence, which is required for the termination of the transcription. In this, basically this sequence is isolated from Streptococcus pyogenes. So this is the Streptococcus pyogenes terminator sequence. What you what is very important in this is this, that the base pairing region should be 20 to 25 nucleotide long only, and your dCas9 handle should contain the hairpin loop structure. Otherwise it will not bind to the PAM sequence. So these are the things which are very important, and your base pairing region should follow the transcription start site. What does it mean? That upstream of the base pairing region there should be the transcription start site. So how we can do the cloning of the sgRNA? If you suppose have multiple sgRNA, how will you do? You can use the BioBrick cloning. In the BioBrick cloning, what happened, you are using the two restriction endonucleases which are having the same compatibility ends. For example, if you take the SPE1, it is producing this sequence, and if you are using XBA1, this is producing this sequence. Here you can see GATC is complementary to CTAG, so it will combine with each other. But now what will happen, this particular sequence ACTAGA will not be cleaved by the SPE1 or by the XBA1, because SPE1 require ACTAGT, but here you are having ACTAGA, so it will not be recognized by SPE1. Similarly, for XBA1 you require TCTAGA. So in this case also you can say T is not here, so TCTAGA, so it will not be recognized by the XBA1 either. So therefore the scar sequence, which is here denoted by the yellow spot, is going to be produced. And this technology you can use whenever you are doing the multiplex cloning or multiplex sgRNA you are having. And similarly, the other restriction enzymes which you can use is VAMH1 and the VGL2. So here you can see the VAMH1 is producing this particular sequence GCCTAG, and here this is producing GATCT and A, and when, because they had the complementary sequence, you can see GATC is complementary to CTAG. So again this is going to be produced, they will combine because they have the compatible ends, so they will combine now, but this particular sequence will not be recognized by BAMH1. As you can see, GGATCC is the sequence recognized by BAMH1, but here you are having GGATCT, not the C, so it will not be recognized by BAMH1. Similarly for the BGL2, if you will see, it has the AGATCT, so A is not here, so AG8, instead of A there is G, so it will not be recognized by the VGL2 either. So this is known as the bio-brick cloning, and this is the bacterial CRISPR system, in which this is the base pairing region, this is the DCAS9, and this is the terminator sequence. And if you want to mutate this base pairing region, you can use the inverse PCR. And this is the bacterial DCAS9 plasmid, which you can purchase online by some vendors. This is an example, good example how you can do the multiplex cloning of the many sgRNA. Suppose this is the sgRNA, suppose you want to target multiple genes, how will you do that? So this is the sgRNA2 molecule, this is the sgRNA1 molecule. Here you can see they are cutting here, they are using two restriction enzymes, EcoR1 and BAMH1. So you will be having this particular portion, which will be having the BGL2 site here, this sequence, and this is the sgRNA2 sequence, followed by the BAMH1 restriction site. Similarly, this particular plasmid you are doing the restriction by EcoR1 and BGL2. Now, as I have explained before, BGL2 and BAMH1 have the compatible ends. So what they will do, this sgRNA2 is going to be brick here, but the scar is going to be produced, because in that case this particular portion is neither is going to be recognized by BGL2 nor by the BAMH1. This is the bio-brick cloning, and similarly you can use another sgRNA in this, and that's how you can do the multiplex cloning. And this is the same thing which is going to be explained here. We are using the BAMH1, we are doing the restriction here by the VGL2 and the NST1, and here you are doing the restriction with the BAMH1 and NSI1. Because the BAMH1 is having the compatible ends with the VGL2, these are the sgRNA which are going to attached with each other, but there will be the formation of scar sequence. So this is the similar to the CRISPR interference. There is another technique which is the CRISPR activator, which can activate the transcription process, and in this case the transcriptional activator which is commonly used is the BP64, which can recruit multiple transcriptional factors, and therefore it can activate the transcription process. In case of the CRISPR interference, you can use the KRAP repressor, which modify the histone proteins and therefore form the heterochromatin formation, and that is the region it is repressing the transcription. As you know, histone deacetylation, whenever the histone deacetylation occurs, because DNA molecule is negatively charged, then histone is going to be deacetylated, and therefore the positive ions on the histone proteins and the negative ions on the phosphate of the DNA molecule, they will interact with each other, and there will be the transcription, there will be the gene expression. So if there is any histone modification, and if you suppose you are doing the modification in such a way that the deacetylation is going to be blocked, then in that case DNA and histone proteins will not interact with each other, and there is the formation of the heterochromatin, and therefore it will lead to the transcription repression. And if you want, I can explain it here as well. So for example, this is your histone proteins, and if suppose, because they are deacetylated, because acetylation is acetyl group is negative, is having the negative charge. So if there is a deacetylation, then it will be having the positive charge. Okay, and because your DNA, you know, the DNA is having the phosphate group, so it is negatively charged. So this positive charge and this negative charge will interact with each other, and there will be the gene expression, and that is the reason HDAC inhibitors are basically used. HDAC inhibitors will block this deacetylation process, and therefore the DNA and the stone proteins will not interact, and the gene expression is going to be repressed. And this is the thing that is done by the KRAB repressor. They modify the stone proteins. If suppose they modify the stone proteins in such a way that the deacetylation is going to be blocked, then the gene expression is going to be blocked as well. So what are the factors which affect the efficiency of the CRISPR? Is location of the targeted sequence, means whether you are targeting your promoter region or the coding region. Length of the sgRNA, which should be typically 20 to 25 nucleotide long, and the PAM sequence. If suppose the PAM sequence is very common, then you will be having the off target effects. So what is the difference between CRISPR and CRISPR-Cas9? In the CRISPR, the major difference is this, that instead of Cas9, what you are using, basically you are using the dCas9. What you are using in the CRISPR, you are using the dCas9, which is the dead Cas9. What happened in the dead Cas9? Both of the nucleus domain is mutated. So it can recognize the PAM sequence, but it cannot cleave the DNA template strand. Okay. And if you have not watched my previous video, please watch this video so that you can understand better about the dCas9. So coming back to the difference, it does not change the DNA sequence, and dCas9 is catalytically inactive, but the CRISPR-Cas9, it can cleave the DNA strand. And again, the CRISPR is the reversible process and the inducible process. If you delete the mutation of the dCas9, it is going to be reversible. However, the CRISPR-Cas9 is not reversible. CRISPR in theory can be used for targeting the promoter, transcribed regions, or any distal regulatory sequences, and CRISPR-Cas9 usually works better whenever you are targeting the exon sequences, and CRISPR is efficient and the specific. So whenever you're designing the sgRNA, the two things which you need to understand, if the sgRNA you are designing against the coding region, the coding region which you should target is at the 5' portion of the gene coding region, or the 5' untranslated sequences. Similarly, the promoter sequences should be there. These are the things which you can see here. This is the thing. So this promoter sequences should be there. Either you target the coding region or you target the 5' UTR region. If you are targeting that coding region, it should be at the 5' portion, and it should also contain the transcription factor binding site. So these are the things which you need to know before designing the CRISPR or sgRNA. So that's all about this lecture, and if you like the content of this lecture, please subscribe my channel. Thank you so much.